Hello, everyone. We'll be getting started shortly. I'm just going to keep an eye on the waiting room. Today shouldn't be a very particularly long or demanding session. The focus is going to be DNA and DNA replication. If we get time, I would like to go back to some of the stuff that we haven't done. So I know last week we were meant to do adaptive immunity. So if I can get back to that, if I have enough time at the end of the session, I would try and get it back to that. But yeah, as usual, just kind of letting me know if you've got any questions, that would be amazing. And I'll keep an eye on the chat. We'll get started in about 5 minutes, hopefully. Again, should take just a couple of minutes to sort out. Right. Let's jump into it. As I mentioned to the people who were here earlier, this is not a particularly long topic, so we might finish slightly earlier if possible. Again, it's just all to do with recording lengths and stuff like that. But today we're going to be talking about the structure of nucleotides, the DNA function, DNA versus RNA, DNA replication. So starting with the basic types of nucleotides or nucleic acids, we have two main branches. So we have DNA and RNA. Now, DNA's function is to store genetic information. So it contains all the instructions for an organism's growth and to execute necessary daily functions. RNA, there's a couple of functions. RNA is mainly used generally in the protein synthesis form, in the protein synthesis arm, and you have different types of RNA. So you have mRNA, which is transferring genetic information from DNA to the ribosome, tRNA, bringing the correct amino acid, rRNA forming the ribosomal subunit structure, They're structurally quite similar to DNA in the sense that they are both polymers made up of nucleotides. So because of this, both are classified as nucleic acids. And the key function the key structural connection of all nucleotides is the presence of an inorganic phosphate group, a pentose sugar, so pentose 5-carbon ring sugar, and a nitrogen containing organic base. Can you think of, based on this key definition of a nucleotide structure, can you think of another molecule that is considered a nucleotide or is considered made up of nucleotides? ATP, yeah, perfect. So ATP is also ADP, ATP, yeah, all of those are also considered nucleotide based because they contain all the basic parts to a nucleotide: phosphate groups, pentose sugars, and nitrogenous bases. So there are loads and loads of different types of nitrogenous bases. You can have adenine, guanine, thymine, and cytosine, as well as uracil. Uracil only present in RNA, not present in DNA. And thymine, again, yeah, therefore T is only present in DNA, not in RNA. And structurally, you have purines and pyrimidines. So can you tell me the difference between a purine and a pyrimidine, anyone? Number of carbons? It's mainly to do with the rings. So purines have 2 rings, whereas pyrimidines are single ringed. Yeah, pyrimidine are 1 ring. And purines always bind to pyrimidines, and that's basically the principle of complementary base pairing by hydrogen bonding. So adenine will always bind to thymine or uracil, and guanine will always bind to cytosine. So as you can see, pyrimidines are single ringed, the purines are double ringed. And this is kind of the crucial differences between DNA and RNA. So DNA is deoxyribose, RNA is ribose sugar, and DNA is made up of A, T, C, and G, whereas RNA is made up of A, U, C, and G. There are other structural differences. For example, RNA is single-stranded, DNA is double-stranded. But those are more to do with the actual polymer itself rather than the single nucleotides. In terms of key differences within the nucleotide, ribose versus deoxyribose and uracil instead of thymine. So when a nucleotide gets a phosphate group added to it, it becomes phosphorylated. And phosphorylated nucleotides, or ADP or ATP, are basically the key parts of energy structure. So they are important in giving us energy, and they're the universal energy currency in the sense that all organisms use ATP as the immediate energy source. Okay. So the next thing we're going to talk about is polynucleotides. So polynucleotides are basically really, really long chains of these nucleotides, and they are held together in 2 ways. So they're held together by a phosphodiester backbone that is formed between a condensation reaction from the phosphate group and one of the carbons on the sugar ring. And because it's a condensation reaction, water is therefore released as a byproduct. And these are phosphodiester bonds, and they're covalent bonds, and they create a polynucleotide chain. These chains have a sugar phosphate backbone, which arises from pentose sugars and phosphate groups. And these phosphodiester bonds can be broken through hydrolysis reactions. And so this is exactly like, I think when we talked about fats and we talked about carbohydrates and we talked about proteins, this is the exact kind of principle behind that. Yep. Any questions so far? Can we have the pattern? No, unfortunately, I can't give you the PowerPoints. The PowerPoints are copyrighted to MedicMind. Do we need to know which carbon in particular is bonded? No, you don't. You just need to know that it is one of the base carbons. I believe it is carbon 4 that's bonded. Correct me if I'm wrong, but it is carbon 4 as you go around the ring. But yeah. Okay. Orientation of a DNA chain. So this is to do with how DNA, it's very important in the DNA function, is the way DNA is orientated. Basically, DNA always runs from 5' to 3'. Now, there are two strands of DNA and they run in opposite directions. So these strands are called anti-parallel. And the reason is because they're parallel, because they're running in the same direction, like they're running in the same kind of orientation, but they're anti-parallel because they're running in opposite directions. But the starting point of a single strand of DNA is known as the 5' end. The end point is the 3' end. And so these two chains run alongside each other and are anti-parallel. Hydrogen bonds hold these together and they form a complementary base pairing structure. So the crucial thing to note is that C to G is 3 hydrogen bonds, A to T is always 2 hydrogen bonds. Sorry, I've got a question here. Why does RNA have uracil instead of thymine? I think it's to do with thymine's structure. There's a lot of speculation about this, but I believe it has something to do with a thymine being quite large, and it's unable to then leave the nuclear pore as easily as uracil is. But I'm not 100% certain on it, and I don't believe you need to know that. Adenine forms a complementary base pair with thymine and guanine with cytosine. And this then wraps around each other to form a double helix. Remember, purifying DNA can be done through a precipitation reaction. So can anyone explain to me the steps of the precipitation reaction? Yeah, like from fruit, like the kiwi experiment. I'm sure you guys have all done that. So I want to see if anyone can remember how to do that step-by-step kind of process. And then we will talk through it if you can't remember. Ethanol is used as a solvent. Detergent to break the membrane. Yeah. So the first thing you do is you add detergent and salt. So detergent breaks the lipid membranes and salt will neutralize the phosphates. Then you add heating to denature enzymes as well as breaking down DNA. You then add protease to break down the histones. And the last thing is ethanol, because DNA is not soluble in ethanol, adding ethanol to the solution allows the DNA to precipitate on top and be separated from the rest of the cellular material. Yep. So this is a summary of your process, and I would recommend writing this down. Oh, can I go back to the previous page? Yeah, I can. So we'll start from here if you want to take these into your notes, and then we'll go back down and through, and just so you can write everything down. Do you need to know this whole process? Yes, this is one of the practical experiments that you'll need to know for pretty much every spec. No, salt is used to neutralize the charge on the phosphate. Never done this experiment. Some people won't have done it yet. It depends on where in the spec this comes up. I think most specs are required to know this, though. It is quite important. Okay. Has everyone got that down in their notes? Okay, this is probably the most crucial part of what we're talking about today. It's the process of DNA replication. It's quite challenging, so I will try and take this slowly and try and explain each step. So the first thing we need to know is, why does DNA replicate? And DNA replication is a key part of the cell cycle. So if you can imagine the cell cycle, let me draw this out. You guys have seen the cell cycle before. You have G1, S, G2, and M. In this S phase of the cell cycle, that's when DNA replication occurs. And DNA replication is crucial to allow mitosis to happen. Mitosis is cell division, and both of the cells need need DNA within their nuclei. So DNA replication is crucial for allowing cells to replicate and genetic continuity to be allowed, which is passing on the DNA. So this process, we're going to go through it in quite a lot of detail. So the first step of the process is unwinding the double helix. And this really, because we want to use both strands as a template strand, we need to break the double helix first. And we do this using an enzyme called DNA helicase. So DNA helicase breaks the hydrogen bonds, allowing for the DNA to split. Remember, the entire DNA does not unwind fully at a time, because that would be dangerous. Because if we unwound the whole DNA, that creates parts of really open exposure, which increases the risk of mutations. So in order to do this, what we do is we do this quite slowly. We do DNA replication at a small area, then we move on and we do it in another small area, and basically in the end, it ends up splitting. So it's kind of, if you imagine like this going in and another one kind of going like this going in, and they keep doing that as they split along that axis. How does it increase the risk of mutation? This is to do with one of the key principles of mutation. Basically, one of the key principles of mutation is the idea that if DNA has enough time that it is not bound to another hydrogen bond, something else can come and bind to it. And that is a mutation, right? Something else that's not meant to bind to, binding to it. And that can only happen when the DNA is exposed. So the more time we spend, the more time we have an unbound double helix, the more risk of mutation from there. Okay, so the next part of DNA replication is semi-conservative replication. So semi-conservative, semi meaning half, or some parts X and some parts Y. Conservative meaning it's not changing. So the principle of semi-conservative replication means that basically one strand is newly generated and one strand is from the parent strand. So free-floating nucleotides that are always in the nucleus anyway, they come along and they bind by hydrogen bonds to this exposed DNA strand that we've got. And because they're only hydrogen bond currently, we need to make that stronger. How do we make it stronger? Well, what we can do is we can form condensation reactions to form the phosphodiester backbone. These condensation reactions are catalyzed by a DNA polymerase enzyme, which reads the nucleotides and enables them to join. Now, as a bit of an A-star level kind of discussion on this, there is a little bit more to this. So remember how earlier we talked about 5' to 3', right? Yes, everyone remember that? We said that basically DNA tends to like running 5' to 3'. Sorry, that was a horrible arrow. DNA likes running in this direction. It does not, so it likes running like this. It does not like running like this. And basically, DNA polymerase is the same. DNA polymerase likes working on a strand that's 5' to 3', not a strand that's 3' to 5'. So for the strand that's 3' to 5', what happens is DNA polymerase works discontinuously. So it doesn't work fully all together at once. It works in kind of fragments. And these fragments are called Okazaki fragments. So they're small bits of the phosphodiester backbone that have been joined up and bits that haven't been joined up. And DNAligase then is another enzyme that can come along and basically it joins these Okazaki fragments up. Yeah. So the strand that works continuously, so the 5' to 3' strand, the one that works normally, is called the leading strand. And the one that works discontinuously is called the lagging strand. And then DNA polymerase also has a proofreading function. Now, this is only in humans. DNA polymerase only has proofreading in humans. It does not have as much of a proofreading function in viruses or in bacteria because they replicate so quickly that it doesn't have the time, which is why drugs that cause mutations in bacteria and viral DNA are really, really effective because they don't have to kind of think about this. Okay, someone's raised their hand. I don't know how to, is there anything that they want to say?
Yes.
Hi, what's up?
Do I have to put video on as well?
No, it's fine.
All right, cool. I just wanted to ask, I joined the class a little bit late, so I just wanted to ask if this is all recorded so I can watch it back later on.
Yeah, all recorded will be put on YouTube at the end of the session.
All right, thank you very much, yeah.
No worries. So remember, DNAligase is the enzyme that works on the lagging strand, and it works in that discontinuous way. Okay. Does anyone want me to go back to the process? And/or summarize it again?
Yes, please.
Okay, give me a sec. What I'm going to do is I'm going to share with you guys a document. Oh, sorry. Oh, I think it's on this screen. Yeah, I'm going to share with you guys a document that's got a lot of, I'll share it with you on the group chat as well at the end of this. But this has got all the processes for the A-level biology spec. It's designed for OCR because that was the spec I did, but it's got them all summarized. And I will take the process of DNA replication from here and just put it on a whiteboard for you guys, so you guys can have that process kind of summarized for you. So give me a sec, sorry. Just going to try and work this out. In the meantime, if there's any other questions, please let me know. So you guys are more than welcome to take screenshots of this because this isn't copyrighted or anything. But this is kind of the exam style step-by-step how to approach this. So normally if this comes up, it's usually a 5 or 6 mark question. And the way that you would approach this is you would literally get a mark for just calling it semi-conservative. You get a mark for naming the enzyme, you'd get a mark for explaining the structure. You tend to get a mark to talking about the base pairing or complementary, forming new hydrogen bonds, marked for DNA polymerase. And then all of these are in the AVP section, which is basically a couple more marks that are for students that are going a little bit above and beyond. So you just need to be, it's not going to be necessarily tested, the Okazaki fragment stuff, but it is good knowledge anyway. So you're all more than welcome to screenshot that process if you've got it. Keep it up for another couple of seconds. Then we've got about another maybe 10-15 minutes of conversation about RNA and RNA structure. And actually, no, there's only one more, which is on structure of RNA. And then basically, can I send the PDF on the group chat of this? Yeah, so I will send this entire document on the group chat. Can you get more than one AVP mark? It depends on the spec. I think some specs do allow you to get a couple of AVP marks. OCR didn't. When I did it, there was only one mark for the AVP stuff. But generally, if you write anything from AVP, you're already having written enough to get the marks anyway. It's just a sort of an added detail bit. Yeah, so we all good for this? Cool, I'm gonna take this back off then. Structure of RNA. So, as we know, RNA is a single-stranded polynucleotide chain. It's made up of RNA-specific nucleotides, which are slightly different, as I'm sure you know this. RNA has ribose instead of deoxyribose. It has uracil instead of thymine. So we know that the key differences, as we discussed earlier. RNA structure, components, and bonding. So this is the table that you want to, oh, sorry, that's on me. Okay, we'll go back. So this was basically the stuff that we talked about earlier. RNA uracil instead of thymine, ribose instead of deoxyribose, basically the kind of general stuff that we discussed in the last one, but now this is more specific, and RNA is one polynucleotide chain compared to DNA is two, so therefore RNA is single-stranded compared to DNA is double-stranded. RNA has RNA nucleotides, DNA versus DNA nucleotides, and that's basically the ribose sugar AUCG. RNA is also tends to be shorter because RNA is for specific proteins. And yeah, so just understanding that these are the crucial differences between RNA and DNA. You've got sort of similar bases, but you've got kind of differences. Types of RNA. mRNA is responsible for passing on genetic information from DNA so that proteins can be made. And so this is done by mRNA. mRNA is made in the nucleus in the process of transcription and travels to the cytoplasm. This is big. DNA is too big to move out the nucleus. Again, mRNA is sequenced in things called codons. So codons are a sequence of basically three bases, which code for each amino acid. Another type of RNA in the cytoplasm, transfer RNA, is in this clover leaf structure, which has an anticodon that is complementary to whatever the codon is going to be. And that is attached to an amino acid. And that amino acid brings in the structure. It brings in the kind of makes the primary structure of the protein. And then ribosomes are made up of ribosomal RNA. And that helps form the peptide bonds in the proteins. Cool. Last couple of things just about structure of DNA, some key terms that you might want to be aware of. So DNA is considered degenerate. Degenerate means that it is there is more than one codon for each amino acid. And that's good because that helps again protect against mutations, because it means that even if you do change an amino acid in a substitution, probably isn't going to affect the protein that's formed. That's just a couple of, that's another key term that you want to be aware of. Right. Any questions? We've gone through a lot quite quickly there. I did earlier say that this is probably slightly shorter generally because there's not as much to it. You sure? No questions? Anyone's got anything they want to clarify with me? No. Okay, so what would you guys be interested in doing for the last 20 minutes? We can do just a couple of things. I've thrown ideas out. We can do things on exam technique and revision, like how to revise and how to get good grades in biology. We can do something on, we can go back and do adaptive immunity. It's kind of fully up to you guys what you're looking for. Exam techniques, I've got one answer saying. Yeah, a couple of people are talking about exam technique. Adaptive immunity. Okay, so what I'll probably end up doing is I'll do exam questions and technique. We'll probably end up doing that now, and adaptive immunity as I will come back to you guys in January, if that's okay with everyone. Cool. So I'm gonna, give me a sec.